Yeah, hello, good evening. We are group 17, and we are presenting on stabilized antigen for improved HIV vaccine. Please, the next slide. So why HIV? Or why this topic? HIV is a human immunodeficiency virus that affects humans and attack the immune system. So HIV was first discovered in 1980s, and it's been 44 years now, and there is no vaccine for HIV. This is because of a lot of factors, and we are going to discuss them. So since the discovery of HIV vaccine, since the discovery of HIV virus, there's no treatment for it. And this is because hello.
Yeah, you can go on.
Okay, please can we start, or it's not possible?
Just go on from where you are now.
Okay. So since the discovery, or since when HIV was first discovered, there is no vaccine for it. And this calls for people are still researching on HIV vaccine, but then there are improvements. There's only treatments which is available, but these treatments are expensive. And also they are limited to certain countries, and it needs to be addressed. So what is the problem right now with HIV? Please, the next slide. Yeah, so HIV is a major global challenge, and it's affecting millions of people. We can see that from this statistics that was made in 2021, the new infections that were discovered were 2 million, and this is for only 10 African countries and half affecting the world. Please, can you move to the next slide? Yeah, so discoveries has been made to produce HIV vaccine, but then it is inactive because there is high variability of the virus. That's the virus has a lot of subtypes, and these subtypes have a high degree of genetic divergence, and therefore it is very difficult to develop a vaccine for it. Also, there is difficulty in eliciting a strong and broad neutralizing antigen response. And the last one is high mutation rates. We know that HIV is a virus of the immune system, and the virus is able to mutate to evade the immune system. They also destroy the T cells. This is the reason why to propose a synthetic biology. We are going to propose a synthetic biology solution to this problem. Thank you.
Okay, next slide, please. Okay, so just like my team member said earlier, one problem with creating a vaccine for HIV is the inability to get an antigen that can elicit a broad neutralizing antibody. And our solution intends to use certain protein engineering technique, directed evolution to be precise, to come up with protein variants that are capable of eliciting a broad immune response. Now, for this solution, our gene of interest is the ENV protein of the virus, and the chassis we will be looking at is E. coli. Next slide. So, for solution, we start with getting a wild type sequence which encodes the outer subunit of the virus, which is the GP120. So once we get that sequence, one of the things we try to do is to introduce random mutations at that sequence using error-prone PCR. Now, this is made possible by the use of a low-fidelity polymerase. At the end of this PCR reaction, we would end up having various PCR products in which certain random mutations has been introduced at various points of our gene of interest. Now, once you have our PCR products, the next thing we would have to do is to, once we have our PCR product, next thing we have to do is we have plasmid of interest, which in our case could be PET19B or PET23B. And this library of sequences that has been generated from error-prone PCR would be introduced into a plasmid, and these plasmids are then introduced into E. coli cells, where they get transformed chemically. So once these transformed cells are gotten, we then induce them to make. So at the end of the day, we would have at the end of the day, we would have various proteins, various variants of GP120 protein, and these proteins are then screened. These proteins are then screened to see how well they bind with antibodies and how well they are able to elicit a broad immune response. Now, once we are able to get maybe the top protein variants that are able to elicit a broad immune response, one of the things we want to do next is to compare them with the wild type protein and see how they compare. Now, depending on the results, this protein variants or the top performing protein variants can now they can now undergo this. We cannot go through this iterative process again, or we can further do some re-engineering again to further improve the activity in terms of how well they elicit an immune response. Thank you.
All right. So when we're looking through the advantages and disadvantages of this approach, we've gotten a few we've gotten a few advantages to weigh our options. And for the advantages, we have a stabilized antigen conformation and an improved immunogenicity as well, and a scalable production. Lastly, is an iterative optimization of the approach we're using. And for these advantages, there's the complexity of the protein engineering, potential immunogenicity challenges as well, potential immune evasion of the virus, and the scalability and manufacturing challenges as well. For the chassis we intend to use, we intend to make sure that the yield is maintained overall throughout each process that we carry out, and there's no possible challenges encountered. So in comparison with other solutions, current solutions, in comparison with our solution, we had two solutions we looked at. The first is the trimerization domains and the mRNA technology. So for the trimerization domains, there's an introduction of trimerization domains and the sulfide bonds between the GP120 and the GP41 and the subunits of the envelope protein. So for this system, the modification helps to maintain the trimeric structure of the envelope protein, enhancing a better stability and immunogenicity. While for the mRNA technology, it involves a modified RNA technology, which involves the introduction of stabilizing mutations into the mRNA coding the envelope protein of interest. Yes. So this helps to increase expression levels and immunogenicity. Okay. Yeah.
Biosafety risk and associated solutions. Biosafety risk has to do with protecting humans, animals, and the environment against risks associated with biological research.
Immune response, which has to do with stabilized antigens, provoking these immune responses, which can lead to adverse effects. Second, you have accidental release. The accidental release of genetically modified organisms used in antigen stabilization can affect or have negative impact on the environment. Thirdly, we have
Manufacturing
contamination during
processes
manufacturing.
compromise vaccine
Manufacturing
safety
processes
efficacy,
vaccine
which
safety
makes it
and
one of our
efficacy,
safety
which makes
activities.
one of biosafety.
Cross reactivity.
And the last point is cross
Life
reactivity.
antigen
Life antigens may cross react with other proteins, leading to unintended health issues instead of actually solving the problem. Biosafety solution.
Biosafety solutions for
Biosafety
unintended
solution
immune responses. One of
for
the solutions
unintended
that we could use
responses.
is conducting preclinical testing and clinical trials to
One of the
monitor immune
solutions that we
responses,
use
and also
conducting
conduct
preclinical
screening
testing
processes
and
to
trials to
identify
monitor
and
the
solve
immune
potential
responses.
adverse
And
reaction
also,
early in the developmental
identify
phase.
potential
For
early
for implementation of for accidental release, implementation of containment protocols in during research and in the facility use
implementation
is
of
required.
for
Also, we have to ensure that compliance with guidelines
during
for
research
handling and
and in
disposal
facility
of the
required.
genetically
Also, we have
modified
to
organisms
ensure that
is
compliance with
properly done.
guidelines for handling and disposal
For
of
the containment during manufacturing processes, manufacturing
manufacturing
practices and
practices and
processes are
processes are
important,
important,
where
where
quality
quality
control
control
measures
measures
should be
should be
implemented,
implemented,
and also the need to adhere to good manufacturing practices
and
is also of importance.
the need to
Well,
adhere to
lastly, for
manufacturing practices.
cross-reactivity, it is very, very important that a
One
screening
that
is done, which incorporates
very
thorough in vivo and
important
in
screening
vitro testing to
done
assess immune response specificity, and bioinformatic tools could also be used to predict and screen the potential for cross-reactivity. Thank you.
Okay, thank you. I'm going to take you through the review and the conclusion. So from our project, we go up for the problem statement, which was the lack of effective vaccine. So from the effective vaccine, there is also a problem with the lack of strong and broad neutralization antibodies and high mutation rate. So the proposed solution for our project was we need to engineer the protein and also to bring about high input and correct error-prone PCRs. So for the method we have just using, there are different pros and cons. So for the stabilized antigen, pros and cons. So we need to, among the advantages of this is that there is improved immunogenicity, also scalable product. Also there are difficulties like complexity of the mechanism at all, and the immunogenicity challenges, and some of the manufacturing challenges. Okay. Also, we have tried to compare with other solutions like trimerization domains and the messenger RNA technology, which are other applications which have been used to synthesize the vaccine. And the potential biosafety risk which we can encounter in our project is that there are probably there could be contamination or cross-reactivity, or an intended immune response can maybe sometimes occur. Also, we have tried to sort out the solution for this, like we need to follow the contamination protocol and compliance so that we can avoid contamination. Also, we need to check for the cross-reactivity screening. Also, the manufacturing practices and the processes should be well maintained. Next. So to conclude, through the use of stabilized antigen in synthesis of HIV vaccines, this is among the promising strategy which we can use to which we can use through the protein engineering, and this can help to boost the stability and the response of the immunity toward the specific viral proteins. So this approach could highly lead to the solution of the challenges which are being encountered by different currency vaccines, which can help in offering the significant public health benefits despite all the minimum risk which can be countered. Thank you.
Right. Thank you so much. So this is everyone from the group 17.
And this is our team.
This is everyone from group 17. We want to appreciate everyone. Constance, Adejuwon, Martins, Valerie, Mathew, and Davis. Thank you.